clinical standpoint, the major risk factor for developing C. difficile infection is the use of antibiotics. And we'll discuss why this is and why we think this is true. But there are a lot of other conditions, comorbidities, a number of things that related to medical care that are associated with risk for C. difficile infection. But the epidemiology of C. diff kind of changed. Koch's postulates for C. diff, in other words, the causative, proving causality of C. difficile as the cause of so-called antibiotic-associated colitis, happened in 1977. And at the same time, it was realized that we could use more antibiotics to treat it. But this kind of changed about almost 20 years ago when all of a sudden there was an increase in both the prevalence and the severity of C. difficile infection. And this was associated with a particular strain of C. difficile, depending on how you type C. difficile. Most people refer to this as the 027 ribotype, and it had a variant toxin. And as I said, it was associated with increased incidence of disease in healthcare systems and a greater severity of disease. Some people were talking, as I was talking to the students, and some of the faculty members, the idea of using genomics to trace the development and move of infectious disease. Obviously, we've been doing that a lot in the past year with SARS-CoV-2, but this was done with C. difficile a number of years ago. And it looked like this 027 ribotype started both in Canada and in the United States and spread around the world. This was from my colleagues at the Sanger in Britain. So they were actually kind of focusing on where did the strains in Great Britain come from, and they traced it back to the Quebec outbreak. But I've had a number of graduate students and postdocs who we thought this is a good system to basically be able to study the microbiome, that environment and community of microbes in the gut in terms of a systems biology approach. What are the interactions between the gastrointestinal microbiome and C. difficile? I don't have to tell you guys how we study the microbiome. There's lots of ways to look at the structure, and in particular to look at the microbiota. We can look at sort of who's there using things like 16S amplicon sequencing or metagenomics, fluorescence in situ hybridization. We can look at functional potential through metagenomics and genome sequencing, but we often want to know what are they doing. What are they actually doing in a given site? What's the physiology of this community? I mean, not just what's the anatomy. You can use metabolomics, proteomics, and transcriptional sequencing. And I'll describe some of the work that we have in applying these various approaches to study the gut microbiota in the setting of C. difficile infection. So why are we so interested in the microbiota? Well, they're not just a series of microbes. Each one has their own physiology. And therefore, as a community, they can actually carry out very complex metabolic conversions in terms of both catabolism and bioconversion of like things that we eat, like resistant starch, fermenting in the short chain fatty acids. But they also synthesize a number of compounds, such as bioactive amines and vitamins and cofactors. And they also signal to the host. They can change epithelial responses. They can actually educate and tune the immune system. And all of this, therefore, shows that the host microbe interactions are very, very important for the homeostasis of this entire microbiome, the host and the microbes that live within the gut. So in terms of C. difficile infection, what we think happens is that somehow the normal microbiota in a healthy person's gut who's not been exposed to antibiotics has this function that's called colonization resistance. It resists having additional microbes come in. Well, let me see if I can turn this on this way. Okay. Having different microbes to come on in. And however, if you disturb the microbiota, for example, by giving antibiotics, you can create a susceptible microbiota. When we're exposed to C. difficile spores, C. difficile is an obligate anaerobe, but can form spores that can survive in the hospital environment. These spores receive certain cues. They'll germinate into the vegetative replicative form. That's the form that can produce the toxin that results in disease. Okay. There are some patients who either are asymptomatically colonized or can clear their infection. There are some patients who go on to severe or fulminant disease, and that's where that mortality of about 20,000 people per year in the US comes from. But we can also treat a large number, about 80% of patients with C. difficile infection. We successfully treat them with antibiotics, okay? Actually, it's probably greater than that. It's greater than 90+% that we can successfully treat with antibiotics. However, about 20%, when we stop all antibiotics, that is the antibiotics targeting C. difficile, as well as the antibiotics that may have gotten them in trouble in the first place, they will recur without any additional antibiotic treatment. And they can go through this cycle, having many, many, many cycles of recurrence. And we think that's because even though we lower the numbers of C. difficile, we don't eliminate it. And if their microbiota stays in a susceptible state, when we stop limiting C. difficile with antibiotics, it blooms again, starts producing toxin, and that's why you get recurrent disease. So the nice idea is to come, well, maybe we can restore the microbiota, and we'll talk about that in a couple of slides. So the work on this, I'm telling you about a lot of it, but not all of it, was done by a team of researchers that I had the pleasure of leading over the past 5 years at the University of Michigan and Rush University Medical Center. And the interesting thing here is we had people with all sorts of expertise. We had people who were 100% infection control, clinical infectious disease. We had data scientists in the Department of Computer Science and Engineering. We had genomicists who were in microbiology. We had people who developed methods and were very interested in the structure, function, and assembly of the microbiota. We have translational infectious disease physicians who work between data science and clinical medicine. We have the director of clinical labs, and we kind of all work together to study the systems biology of C. difficile infection. And one of the places that we started was this idea of recurrence, right? These patients are also responsible for a lot of the morbidity and mortality due to C. difficile infection. And we were very interested in this idea that people had of microbiota restoration. So the idea of restoring the microbiota for health is something that goes way, way back, and goes back to this idea of fecal transplantation. The microbiota was thought to be important even in ancient Greek times, when Pliny the Elder would recommend fermented milk products, and depending on how you read these ancient scripts, maybe fecal transplants for treating a variety of diseases. In traditional Chinese medicine, the idea of fecal transplant had been used for a long time. The idea of making so-called a yellow soup, where you would take feces, kind of mix it up in water, allow the solids to kind of settle out, and you took the yellow liquor on top and used that to treat a variety of illnesses. But when it came to some of the more modern use of fecal transplantation, there was a report. Even before we knew about C. difficile, we knew that some patients with antibiotics would develop colitis. Surgeons in the 50s would try fecal transplantation with good success. But as far as C. difficile, there was a lot of attention being paid to the use of fecal transplant for C. difficile in this trial, this randomized controlled trial that came out about 10 years ago, where they had patients with multiple rounds of recurrence and compared directly fecal transplant to standard of therapy vancomycin, and showed that the fecal transplant had a 94% success rate, where standard of therapy vancomycin only had a 30% chance of eliminating recurrent disease in these patients. Interesting to note, at the time, they stopped the trial early because the Data Safety Monitoring Board said, hey, look, this is such a significant difference that it's not fair to withhold fecal transplant to patients who receive vancomycin. So really, it was only 16 patients that caused this change, really sea change in how we treated recurrent C. difficile, and a lot more people started doing fecal transplantation. Such to the point that in the recently revised guidelines for the treatment of C. difficile infection that came out from the Infectious Diseases Society of America, it was a strong recommendation for patients with multiple recurrences of CDI to be treated by fecal microbiota transplantation. They called it strong recommendation, and at that time they said moderate quality of evidence. There were more than just those 16 patients, but still only now have we started doing larger and larger studies of FMT for treatment of C. difficile infection. But let's go back to that time. I pointed out that New England Journal paper came out in 2013. And at that time, I had a PhD scientist who had finished her graduate school at the University of Maryland Medical School. She had learned how to study the microbiota. She was actually studying how the microbiota might influence vaccine efficacy. She's now an assistant professor at Clemson University, has her own lab. But when she came into the lab, I pointed out that there was a group of investigators led by Johann Bakken, former president of the IDSA, and a number of folks in Duluth, Minnesota. And for probably 20 years at the time, they had been using fecal transplantation to treat patients with C. difficile infection. And he had contacted me and he had sent me some samples and we had sequenced them, and I needed someone to analyze these samples and say, hey, what happens when you actually treat a patient with FMT? We assume that we're replacing certain elements of the microbiota, but we didn't know that for sure. So Anna quickly analyzed the data was there, published this paper in MBio shortly after she arrived in the lab. And this is all published, I won't go over it much, but we had patients who had a lot of recurrence. So on this timeline, 12 months prior to the time that they got their FMT, these patients had multiple positive tests for C. difficile by all these pluses, and they had received many, many courses of antibiotics to try to eliminate the recurrence. And like, look at this patient, look at how many rounds of antibiotic therapy they had. But then they received an FMT. We actually got a sample prior to FMT. We got the donor sample. Some of them were missed because the nursing staff wasn't used to saving the donor sample. They usually threw them away, but then they said, oh, no, we need this for this study. And then we got samples about 2 weeks or so after they got their FMT. We did the 16S amplicon sequencing. I'm sure you're relatively familiar with that, but then we kind of processed the data. And we wanted to look at the community structure because that's what you can get from 16S. Now, let me orient you a little bit to this heat map. So each sample is in a column from the patient. The patients are numbered 1 through 15 here. Each sample from a patient is in a column. The samples in red were obtained from the patients with recurrent C. difficile prior to their fecal transplant. In green are the samples after fecal transplant, and the donor specimens are in blue. And we allowed the data to just be reorganized by which communities were most similar to each other. And if you notice, there are two major clusters here. And this one is more diverse. There are many more different types of microbes in there than in these samples, right? All the donors are from this more diverse, complex community type. Most, but not all, of the patients after they received FMT, the green samples, they're also, looks like they transferred things. It wasn't one-to-one with their donor, though. It was interesting to notice that it wasn't like you completely replaced it. They did have their own microbiota, and it was altered and enriched by the FMT, but not completely replaced. Now, in the study, we had 15 patients, and 13 of them responded to the FMT, 2 didn't. And I was pretty excited when Anna showed me this heat map. I go, these 2 patients after that still had a lower diversity that kind of went along with the pre-samples here. I said, were those the 2 that weren't cured? And she goes, no, I thought so too, but that's not true. So structure does not predict function. That's an important thing, and we'll come back to that. And you can't necessarily also diagnose a patient based on sequencing, looking at their microbiota, in spite of the fact that patients will bring in these reports. They spend money to companies that say, oh, we can tell you what to eat and what not to eat and how to take drugs and stuff based on a fecal sample. That's not quite true. Let's talk a little bit about the strategies for FMT. When it was initially done, you would actually have directed donors. You'd have family members or friends that would donate the feces and it would get given to the patient. Now, just because of that, we need to test for pathogens. There are companies such as Open Biome that take feces and screen it for pathogens and freeze it, and they can ship it. And that's what we use for most of our FMT now. There are companies, and we'll come back to this, that are trying to process feces in different ways, not just screen it, but kind of try to eliminate pathogens and enrich for quote unquote good bugs. 2 companies are Rebiotics and Series. But there should be caution with this. When you're using directed donors by accident, sometimes you could do things like transfer drug-resistant organisms that make it through the screening. And when the SARS-CoV-2 epidemic came about, that really did put a squash on all FMT, because the FDA said, hey, wait a minute, we can detect live SARS-CoV-2 in feces, so we better stop doing fecal transplants. They came up with new ways to screen for it and to inactivate it. And so now that company I was telling you about, OpenBiome, is shipping out all of their tested lots, but they're not going to generate any more. So we need to come up with something else for FMT. I mentioned that there are some companies that are trying to process feces a little bit more than just kind of rinsing it and screening it for pathogens. This company's series last month in the New England Journal, they published their phase 3 trial. It's the first phase 3 trial of this, which is enriched for spores. They use a patented process that use ethanol and other things to eliminate pathogens and enrich for the spore forming fraction of feces, which seems to be necessary for treating, and necessary and sufficient for treating recurrent C. difficile, and they had better response. And this was a placebo-controlled trial of this SEER 109 of this processed feces versus placebo. And this is, they'll probably go for FDA approval, and this is causing some more enthusiasm. Just as an aside, their phase 2 trial unfortunately failed, partially because of how they were testing for patients, and there were lots of reasons that they did not reach superiority in that, but they changed that for their phase 3, and they did have a good response here. So how do I study C. difficile? I do study it in patients. One of the papers that was sent out was some of the work that we've done in C. difficile in patients. I won't talk too much about that anymore. I want to kind of go to how we study C. difficile in model systems. So Koch's postulates I mentioned were fulfilled in 1977. That was using hamsters as the model system. Basically, you can take a hamster, give them a dose, 1 intraperitoneal dose of the antibiotic clindamycin, and then you give them a few C. difficile spores, and the animals are dead within 24 hours. Now, one of our favorite laboratory animals is the lab mouse. And around the same time, they tried to give clindamycin to mice and then infect them with C. difficile, and they could not replicate what they found with the hamsters in the 70s. Someone from the University of Michigan, Rolf Frater, who was in the Department of Microbiology, and this infectious disease fellow, Ken Wilson, who's now at Duke, did show that it's not that the mice are resistant to C. difficile, because if you took a germ-free mouse and infected them with C. difficile, they would die. But there was something about the microbes in the mouse gut such that clindamycin wouldn't change them enough so that they'd be susceptible to C. difficile, but hamsters would. So this kind of went on for a while. People would use hamsters, but as I mentioned, people weren't studying C. difficile. Vancomycin and metronidazole were known to be effective treatments in most cases, except in recurrent cases where FMT was sort of brought in. But after that 027 ribotype came out, and we had more severe disease and higher incidence of disease, that prompted a number of people to say, well, let's see if we can come up with a mouse model to figure out what's going on. And a group in Boston published a group where they gave 5 antibiotics followed by clindamycin and could finally make mice susceptible to C. difficile. I thought that was a little excessive. We were looking at other antibiotics and the effect of the microbiota for other research questions, and we decided to use one of our most disruptive antibiotics, cefoperazone. And if you administer that in the water for 4 days, the drinking water of animals, and then orally challenge with C. difficile spores, we would actually have rapid, and using baseline, change in weight as health. If we gave this strain VPI 10463, the animals would die within 2 to 4 days. They'd get very sick, lose a lot of weight, have severe colitis, which I'll show you about. A bit of an aside, VPI, yes, it is named after these strains with the VPI designation are named after the Virginia Polytechnic Institute. The anaerobe lab that was at the vet school really created a lot in terms of anaerobic microbiology. So that's a bit of history of your institution that you might want to look at a little bit. If we used an 027 strain, it would depend on the kind of day that we do it. In some cases, the animals would lose weight rapidly, or in some cases, they'd get sick. A few of them would die, but others would recover. We could use a different laboratory strain that has the same toxin as the VPI 10463 strain, but produces less of it. Those animals would get mild colitis, but would generally recover, and most of them would survive. And if we used a non-toxigenic strain that we isolated from one of our patients, there was no disease whatsoever. Well, what is the histopathologic correlates of this disease? Here's the colon of an antibiotic-treated mouse that received saline as a gavage, as opposed to one that got about 200 C. difficile spores. You see that in 2 days, the development of a lot of edema, a lot of inflammation, hyperplasia. These are the pseudomembranes that you would see endoscopically if you were to scope a mouse, and there's a destruction of the epithelium. So great, we have a good mouse model of C. difficile. So we were very interested. Well, how could the normal microbiota mediate this so-called colonization resistance? Are they competing with C. difficile for food? Are they changing, I'll get into bile acids a little bit, are they changing host products in a way that can inhibit C. difficile? Are they directly producing antimicrobial products? Or are they signaling through the host, either the immune system or the epithelium, to produce antimicrobial peptides or immunoglobulins or other immune responses that limit C. difficile? So how do we do this? So I had another postdoc who arrived in the early 2010s. This is Casey Terrio. She is a professor now with tenure at NC State University. She really wanted to see about this idea of what's the metabolic environment in the gut after you give antibiotics, and how does that influence what's going on with C. difficile infection. And she did an untargeted metabolic approach in comparing animals that got cephalosporin and were therefore susceptible, animals that were never treated with an antibiotic, or what she had found in her earlier studies, you could treat them with cephalosporin if you don't infect them at this time, but allow the microbiota to recover for 6 weeks, they again become resistant. So you kind of suppressed organisms, but you get a different community structure that can still have colonization resistance. And again, this is all published, so I won't go into the details except focus on one of her various results. She noticed a change in bile acids. And it didn't matter how you were resistant. Either you never got antibiotics or you got antibiotics but were allowed to recover. Animals that were resistant to C. difficile had a different bile acid profile than the ones that were susceptible. In particular, she was looking at taurocolate. So bile acids, I'm sure the students have heard about it, but let's talk about it. These are derived from cholesterol. There is a carboxylic acid group on the end here, or the carboxylic acid group is protected by conjugation to the amino acids glycine or taurine here at the R4 position, okay? Or they have other substitutions on the rings. But what does all this mean? Who cares? Bile acids are secreted by the host, and they're synthesized in the liver from cholesterol. And as I said, they can be conjugated. Well, taurocolate here, this conjugated primary bile salt, is actually one of the most potent signals for C. difficile spores to germinate and produce the vegetative form of C. difficile. Okay, what happens to these bile salts, these conjugated bile salts? Well, there are bile salt hydrolases that some microbes can carry that will cleave off the amino acids, releasing glycine and taurine. Interestingly enough, glycine is a good co-germinant with taurocolate. That was shown by my colleague Joe Sorg and Ken Wilson, who I talked before, that taurine in the presence of glycine is an even more efficient germinant. Okay, and some of these deconjugated forms are either positive inducers of germination, or in some cases can inhibit germination. And then how about these secondary bile acids? For example, at the 7 position here, if you remove the hydroxyl group, it takes about 7 steps. It's not just a simple step, but if you have a cluster of genes, you can produce these secondary bile acids, such as deoxycholate. And what Casey has shown in my lab and also is focused on in her independent lab at NC State, deoxycholate is a mild germinant, but more important, it is highly inhibitory to vegetative C. difficile. So what happens when you take antibiotics? Well, at least what we can theorize, and we can show this a bit, is that you eliminate or at least greatly decrease this microbiota-mediated metabolism of bile acids. So you accumulate germinant and you lower the amounts of the inhibitory secondary bile acids. My friend and colleague Eric Paymer, who was originally at the Memorial Sloan Kettering Cancer Institute, is now leading a microbiome center at the University of Chicago. I followed up on work with Joe Sorg, who, as I told you, studied spore germination, and using a kind of informatics approach said, yeah, we should look at bile acids, and found that giving certain kinds of organisms, such as Clostridium scindens, yet another Clostridium species, could actually restore the amounts of deoxycholic acid because it has potent 7-alpha dehydroxylase activity, producing this C. difficile inhibitory secondary bile acid, and showed that, yeah, you could actually restore bile acid mediated resistance in a mouse model. But are mice exactly the same as humans? You know, not exactly the same as we know that their anatomy and physiology is different. How about their microbiota? Well, this is work that goes back a long time ago, almost 20 years ago. You know, this is work from Ruth Ley in 2005, that the microbes in mice and humans are close. They're related, but they're not the exact same species. They're just closely, presumably evolved and, you know, evolved in the symbiosis to be similar classes, but are different in mouse and human. And people have been looking at this. Is the mouse really a good source of information for the human? And we'll get back to that a little bit more later as well. Well, so what Anna and Casey did before they took off to start their own labs, they kind of worked together with some of our gastroenterologists. They got an additional group of patients who were treated with FMT for recurrent C. difficile and showed that, yes, bile acid mediated metabolism was restored in these patients. So not just a mouse model that Eric Paymer showed and that we had shown, also looked at other functions, such as the production of short-chain fatty acids, which are anti-inflammatory and have some inhibitory activity in their own right against Clostridioides difficile. So the final thing that Anna did, Casey left first, Anna says, well, wait, how close are they? Can we use human feces to treat recurrent C. difficile in mice? Well, before we get to that question, what Anna had to do, she had to develop a mouse model for recurrent C. difficile. So what she did is she took our standard model of making animals susceptible with cephalosporin, infected them with C. difficile. And what we're monitoring on the y-axis is the number of viable C. difficile that we can grow out of a gram of fecal contents, okay? So, yep, she infected the animals as normal. We got high levels of C. difficile. She treated them with one of the standard antibiotics we used in an equivalent dose, vancomycin, and showed that indeed vancomycin would move, this is the level of detection, about 10 to the 2 per gram, below the level of detection when you treated with vancomycin. But when you stop the vancomycin treatment, let's look at the yellow tracing here, you saw a rebound. These mice would recur. And we're not sure, we're still trying to investigate. Is this because spores were on the fur? We did change the cages, but do they exist either on the fur or were they kind of hiding out someplace in the gut? Wherever it is, they would be able to recolonize the animals. The animals would get recurrent disease as well here. If we sacrifice some of the animals, they would have a colitis and they would stay colonized with the organism. But if she took feces from a non-antibiotic treated mouse and gavaged it in at least once, or in this case twice, you would get a slight rebound in the C. difficile, but then the C. diff would finally be eliminated. You could kind of unmask it in a certain amount of the mice by giving them a little of clindamycin, but still it would go away once the effect of the clindamycin would be there. This is something that we're also investigating a little bit more. But to get back to Anna's question then, she then took human feces from those patients I reported that had restoration of their short-chain fatty acid, bile acid metabolism. She took the same donor feces that had been frozen and given to the patients and tried to use it to treat the mice, again, using that same model. If you didn't give FMT, this is the pink line now, yeah, the animals would recur as I showed you previously. If you gave them mouse feces, you would eliminate carriage here, as we showed before. Interestingly enough, if you gave human feces, and again, this is human feces that successfully treated a human with recurrent C. difficile, it didn't work. Why is that? Well, she's following up on this. It's not published yet. She's trying to work it out in her independent lab. Only mouse FMT into this recurrent system would restore the bile acid pool appropriately. And she's also looking at the microbiota. It's even though these mice have kind of a antibiotic damaged microbiota, this is an antibiotic damaged microbiota that's composed of mouse adapted microbes. And it appears that the human adapted microbes, even though quote unquote they have good functions, can't compete with those mouse adapted ones. And she's kind of following up on that. So yes, model systems are important, but you got to be careful and understand what you're modeling. So final story, we're going to shift to a non-infectious form of colitis, inflammatory bowel disease. We don't have to talk about the different forms, but this has been called idiopathic. It's not supposed to be caused by a discrete infectious agent, but in this disease as well, the indigenous microbiota as a group, as a whole, are thought to play a key role in the pathogenesis. And a lot of this work has come from the fact that there are mouse models of inflammatory bowel disease. They're either genetic models where you can kind of change the host by knocking out genes, in particular immune regulatory and immune response genes, or you can directly damage the intestine with things like dextrans sodium sulfate and such as chemical. This is more a model of injury repair. Some people think maybe the genetic models are closer to what you find in humans because there are genetic components to IBD susceptibility. But we were pretty interested in this because, the microbiota are important not only for infectious colitis such as C. difficile, but also in terms of immune-mediated diseases like IBD. And there's a lot of discussion among the immunologists that, well, we should be very careful how we model our systems to mimic host responses. And this paper came out a couple of years ago, and it was kind of fun. They referred to so-called wildling mice, where they took laboratory mice that were germ-free and then associated with them with the microbiota from wild-caught mice. And what they found is that this was closer in terms of mirroring response to drugs and immune responses as humans were, as opposed to laboratory mouse with the so-called specific pathogen-free microbiota. And in this paper, I was interested to find out when they did their microbiota analysis that there were a lot of helicobacters in both the wild mice and the wildling mice, that is the germ-free lab mice that actually had the wild microbiota transferred to it. Microbiota have been eliminated from lab mice because they're thought to be quote unquote pathogens. And we'll discuss that in the next one, because in a lot of the mouse models of IBD, they developed from people knocking out immune genes such as the IL-10 cytokine or adoptive transfer into rag mice with certain phenotypes of T cells, or animals that were deficient in T cell signaling. Many of these immune altered mice would develop so-called spontaneous colitis. And it was found that the microbiota would influence how bad the disease were. And the Helicobacter species were found to be a sufficient trigger in a number of these different mouse models here. So people said, oh, we don't want our mice getting sick. This is a pathogen. Let's get rid of it. But as I just talked about, maybe this is because it stimulates the immune system in a particular way that's normally beneficial. So I have a colony of IL-10 knockout mice on the Black 6 background, and ours are free of Helicobacter. There's a low rate of spontaneous colitis. Maybe 5% of the animals, if we hold them for 15 months, will spontaneously develop colitis and die. But if we infect them with Helicobacter hepaticus, within 1 to 2 weeks, we get really rip-roaring colitis. You can get crypt abscesses, you get inflammation all the way through, a lot of hyperplasia here. So, hey, great, we have a mouse model of IBD. And that's what I worked on for my postdoc and as an assistant professor. But then I got involved in C. difficile research, became more fundable, and I kind of left this behind. So I had a postdoc who came in and says, hey, I really want to work on mouse models of IBD. So do you want to bring that back? I said, sure. We got the strains. We still got the mice. She showed very clearly that if you put Helicobacter hepaticus into an IL-10 mouse, 7 to 14 days afterwards, if you monitor fecal lipocalin as a biomarker of gut inflammation, yeah, there's a lot of inflammation there. not present in wild type animals with or without Helicobacter. Okay. And yes, putting in Helicobacter, developing inflammation will change the microbiota of IL-10 knockout mice, but not wild type mice, whether or not they have Helicobacter hepaticus. The rest of the microbiota, and this is only one organism that's present there in a low amount, it doesn't change globally the structure and presumably the function of the microbiota. It had been well known that patients with IBD are more likely to get C. difficile, and when they have C. diff infection, it can be harder to treat and be clinically worse than patients without IBD. So this postdoc, Lisa Abernathy-Close, who's now moved on to a different lab, she wanted to know, hey, if we trigger IBD in these IL-10 knockout animals with Helicobacter hepaticus, are they susceptible to C. difficile? And this was recently published, and I know you guys discussed this paper, so I'll go over this really quickly. As a control, she wanted to do all the controls. She wanted to make sure that IL-10 animals were susceptible if we treated them with antibiotics. Sure enough, if you take an IL-10 animal, treat them with antibiotics, infect them with C. difficile, they die within 2 days, okay? And they have very, very severe disease. But how about if you then take an animal with colitis due to Helicobacter hepaticus, don't give them antibiotics, but challenge them just with C. difficile spores? Yes, they get colonized. They get colonized to a high level, almost as high as if they'd gotten antibiotics. Some of the animals it was high, some of them not as much. So it's not as penetrant, but yeah, in many of the animals, you can get them infected with C. difficile. The animals would have worsened gut inflammation, but they wouldn't die like the antibiotic-treated animals with the same VPI 10463 strain. So that raised a number of questions for us. It's like, okay, it worsens the IBD, which is kind of consistent with what we see in patients, but they don't succumb to the disease. So it's not exactly the same susceptibility as if you give antibiotics. So I will give the students a chance, since you guys read and discussed this paper. Any questions before I tell you what we've been doing next? Anyone want to unmute or type? I don't know. Can I see the chat? I don't see the chat. If someone types it into the chat, I can. Let me pause for a minute. I have some time.
We have a few questions in the Q&A panel, but I think for this sort of intervening like this, students, feel free to type into the chat if you do have questions that you want to get answered right now.
Anything? I can move on and finish off the story in the last 10 minutes or so. So what questions did we have? So when we published this paper, I told you Lisa Abernathy-Close was the postdoc. She'd moved on. The graduate student, Maddie Barron, who had started picking this up, she goes, yeah, I'll take this over. Let's move on that. So some of her questions that she had, and she answered them appropriately, such that at the last week of April, we have approved and scheduled her thesis defense. She wanted to know, well, how important is that inflammation? We know that we can treat patients with IBD in ways to reverse inflammation. Does that change susceptibility to C. difficile? Is it really the microbiota, or is it the combination of the microbiota and inflammation? That's how we wrote it in the discussion of that paper that I was talking about. There's some evidence, both plus and minus. And what are the host responses in the first place that can confer susceptibility, either directly or via changes in the microbiota? So how did she do that? Well, the first thing she wanted to do is like, well, can we treat mice with IBD using an FDA-approved therapy for inflammatory bowel disease in humans? She used the anti-P40 monoclonal antibody, ustekinumab, the trade name for it is Stelara, and it actually targets, is specific for the P40 subunit that's shared between the inflammatory cytokines IL-12 and IL-23, eliminating both Th1 and Th17 responses. Work from Marika Kohlberg and Alan Scher's lab showed that this is more important in the Helicobacter hepaticus IL-10 model, but kind of picked off both with anti-P40. So what did she do? She took IL-10 animals, she colonized with C. difficile, with Helicobacter hepaticus. She showed that yes, they had fecal lipocalin by 2 weeks after that. That was pretty high here. Let's look just at this teal color. The animals, as opposed to their baseline, developed inflammation. And then she gave them the anti-P40 or an isotype control antibody every 3 to 4 days for 3 weeks. And after 3 weeks, if they got the control antibody, there was no decrease in the inflammation. But if they got the ustekinumab, there was a decrease in the amount of inflammation. And that was associated with a decrease in the histopathologic amount of colitis. And she looked at a number of cytokines, but she decreased IL-17 as expected and TNF-alpha as expected. Okay, great. So then what'd she do? She took another group of mice. She triggered colitis with Helicobacter hepaticus. She treated with the anti-P40 monoclonal antibody. And then, again, no antibiotics here, challenged with C. difficile spores and monitored colonization. And she found that, yeah, indeed, treating inflammation restores colonization resistance. Okay, so if you took animals that had colitis, they got Helicobacter hepaticus, and they got the control antibody, and then were challenged with C. difficile, that's the repeat of the earlier published studies. Yep, out of those animals, out of the 20 animals, 15 of them got colonized. Some of them still didn't get colonized or were intermittently colonized. But of the animals that received the anti-P40 treatment prior to being challenged with C. difficile, in other words, we treated the inflammation, only 2 out of the 17 animals were ever positive for C. difficile. Most of them were below the limit of detection. And that was statistically significant. If you treat, you do kind of decrease the susceptibility to C. difficile. But it's also nice, at first, you would say, well, it's not 100%. Anytime that there's variation, you can actually study some biology. If something's always absolute, you have a hard time looking at what might be important because it's either all or none. So what Maddie did next is she looked at the microbiota of these animals. She looked at the baseline. All of our animals are about the same. Great, that's good. How about after she triggered colitis? 14 days after triggering colitis, this is what we showed in the previous paper. Yeah, the development of inflammation changes the microbiota. But if you then treat with the anti-P40 monoclonal antibodies, those in the kind of orange color here, they started changing back. They changed away from the animals that had continued colitis, no treatment. But for the most part, many of them went back. And here's the other time, there were 2 outliers here. Remember, 2 of the 17 animals were susceptible to C. difficile. And lo and behold, it was these animals that had these 2 communities here that didn't change much, despite the fact that they had ustekinumab treatment. Okay. Well, she wanted to look at that even more. Okay, that's great. But remember, we had all the possibilities. We had animals that were treated with the monoclonal that were susceptible or not susceptible. We had animals that had colitis but not treated. Some of those were susceptible to C. diff, but a small handful were not. So we had all the possibilities there. So how do we test hypotheses? Well, she goes, can you buy me some germ-free mice? I said, yep, what are you going to do? I'm going to take all 4 classes, microbiota from all 4 classes of animals, treated and untreated with regards to the ustekinumab, susceptible or resistant to C. difficile based on challenge. And I'm going to collect their feces, okay? And then I'm going to FMT into wild type germ-free C57 black 6, okay? And what did she find? Well, she did the experiment. There were 4 groups. She did it twice. There were 8 groups of animals. And in 7 of the 8, she got exactly what she predicted. If you are resistant to C. difficile, whether or not you were treated with the anti-inflammatory, when you transfer that microbiota to a germ-free wild-type animal, you were resistant. In 3 of the 4 cases, if you transferred a susceptible microbiota into the animals, those animals were all susceptible to C. difficile. And since these were wild-type animals, interestingly enough, they don't develop colitis from H. hepaticus, and these animals died right away because of that highly virulent VPI 10463 strain. So she's now finishing this up. Once I'm off of my meetings today, she gave me another draft of her paper so that we can submit this soon. And she basically wants to know some other things. What are the microbiota changes due to H. hepaticus infection? She's already done the 16S analysis to see what's the difference in all the classes. What's different in the mucosal inflammatory response? She's done some targeted qPCR for gene responses, as well as we've done some just bulk RNA-seq, and we see differences in the different classes. And the key question that we have for going forward that's still not answered, but therefore we have the ability to do more work, really, how does the development of inflammation change the structure and function of the microbiota? Because our germ-free transfer shows that it's not necessary to have inflammation, but the microbiota is obviously changed by the inflammation. So how does that happen? Okay. So let's back up a little bit. What do we do with this whole idea of studying the microbiome? I didn't talk about it, but there are many, many diseases in which changes in the microbiome are associated with either worsening or susceptibility or resistance. Slowly, we're just going from association towards actual causation. And we understand how we might be able to manipulate the microbiota, either intentionally with antibiotics, diet, or how do some of the things that happen, such as aging, I have an R1 on aging in the microbiota, how does that affect the structure and function of the microbiota? And really, we do want to try to manipulate the microbiota in a way that we can improve health. So in this review that I wrote before, I had this kind of idea of how microbiota can fit into this idea of precision health, which you probably heard about. The NIH has this so-called All of Us initiative, where they're sequencing the genomes of, I can't remember, they wanted to have 100,000 different individuals, I think, so that we can do host-centered precision medicine, the Human Genome Project promise, that can we understand by understanding the host genome, can we predict genomic susceptibility to the various diseases, predict drug responses, predict adverse reaction to both drugs and other treatments. But what if we combine that with a microbiota-centered precision medicine, where we look at microbiota structure and function? Can we actually try to combine all of this to have customized therapy for the future? Okay. So all of this work is really team science, as I talked about. I talked about all of my collaborators at the University of Michigan and elsewhere at the PI level, and of course, all the people in the lab with all sorts of expertise that can work together on these kind of team science cooperative agreements, where you bring in people with various expertise to address these very complex problems and hopefully try to learn more about human health and disease. I'd be happy to take any questions now. And thank you again so much for the invitation, and thank you for your attention.
Great, thank you so much. There are a number of questions that came in through the Q&A portion, and students feel free to continue, and other attendees to continue adding to those. I will go through and pick out a few to start with. Kristen Howard asked, do you think there is a potential role for FMT in chronic post-cholecystectomy bile salt diarrhea or chronic diarrhea following weight loss surgery?
Yeah, the whole idea, both in terms of obesity directly, plus all the things that we do, and people who have dumping syndrome and all sorts of the post-gastrectomy syndrome and all that. It might be FMT, it might be as simple as FMT, but one thing that should be noted, a lot of people are looking at FMT to treat a variety of diseases, and they followed the procedure that was originally done with feces, where you take the feces and you put it in a wearing blender and you put in sterile saline. I don't know why, you don't need sterile saline, whatever. Whip it up in the wearing blender and give it to the patient. And I always pointed out, I said, I wonder if you need some of the 99.99% of the microbes that you kill through that treatment. And some people go, what are you talking about? I said, there are a lot of anaerobes that you destroy by putting them in a wearing blender. Needless to say, you're destroying a wearing blender. And so, yes, it can work there, but some of these bile acid transformations are carried out by spore formers that survive that. But some of them also are carried out by very oxygen sensitive organisms. So we need to be careful. Might not be FMT. That's why companies are trying to isolate individual organisms, characterize them genomically and physiologically to try to put together rationally designed consortia to treat specific things, depending on what you want them to do. I mean, we're almost to the level of oh, patient has cancer, let's give him chemotherapy. Well, which one? Chemotherapy. Well, we would never say that. You know, do you need an alkylating agent? Do you want it on and on and on? Hopefully we can get to that level of specificity and rationale with manipulating the microbiota.
Great. And to follow on a little bit with sort of translating this to other disorders. So there's a question from Anis Sarkeesian. How do gut bacteria maintain low levels of the yeast candida albicans in our gut? And is there any evidence to connect the obesity epidemic to long-term loss of gut microbiota?
Yeah, the fungi in the gut, Candida albicans and other Candida is important. It's, you know, they're relatively low levels in terms of numbers, but they can be quite important. And there are a number of people here at the University of Michigan who are looking at that. And it's not clear what those, we don't know. How is it? Is it things like competing for metabolites? Is it producing things like short chain fatty acids that are not as beneficial for Candida? It is true. One of the observations is that people who are given antibiotics can have Candida overgrowth that can lead to Candida vulvovaginitis or just Candida on the skin and other places. So don't know about that. And the obesity epidemic and the microbiota is an area of very, very, very active research. And the answers aren't clear. There is some sort of relationship, but we're not able to really either predict right yet, or certainly not able to treat, quote unquote, treat obesity by targeting the microbiota yet. Sort of on that idea, someone had asked about supplements with probiotics and prebiotics. Again, it depends on what they are. Probiotic, there's so many different ones, and most of them are derived from fermented foods. Many of them are lactobacilli. Some of them are bifidobacteria, not all of them. But no one's coming up with any of the obligate anaerobes that are normally present in the gut as probiotics. They are now, but the traditional ones are not. Prebiotics, you know, the idea of giving things like resistant starch, that is actually being tried. There's a trial going on here at the University of Michigan in patients undergoing allogeneic stem cell transplants who are at risk for developing graft versus host disease, seeing if we give them something that might increase the amount of fermentation to butyrate, that anti-inflammatory beneficial short chain fatty acid, would that limit the amount of GVH? And there's some preliminary results in there.
Great. And then I want to make sure we ask Katie Burgess's question. Have there been any clinical studies on the rate of co-infections of Helicobacter pylori and C. diff in human patients? Given the high prevalence of H. pylori infections and that many cases can turn symptomatic and cause chronic inflammation, it would seem that this might be a way to translate into human population.
Yeah. So interestingly, even though they share the same genus name, Helicobacter pylori, Helicobacter hepaticus doesn't live in the stomach like pylori does. It lives further down. And pylori, interesting, has a whole different set of genes that allows it, that tunes its interaction with the host immune system that it does not share with Helicobacter hepaticus that lives, as I said, in the lower GI tract and in the liver, hence its name. In fact, Helicobacter hepaticus shares some genes that we're looking at, like that cytolethal distending toxin that was present in that first paper. That's actually shared with the related Campylobacter jejuni, which is a completely different genus, but lives the same way. So again, you're closely related genetics to people that you're related to, like Helicobacter and Helicobacter, but some of your accessory genome is determined by how you live your life. So therefore, Hepaticus and Campylobacter jejuni share certain kinds of adaptations there. So, yeah. Oh, the cat. Cat's name is Cloudy. Yes, the cat's name is Cloudy. And yes, during the time that I was locked at home with Cloudy, I did think it was kind of surreal. So every so often I did kind of play with Salvador Dali. So yeah, thanks. Thanks for the questions. Are there any more? I know we've reached the top of the hour.
I think that the ones that remain, you sort of already answered. So unless anybody else has anything they want to jump in with at the end, thank you so much for sharing just a wealth of research, and I appreciate you taking the time to both talk with the students and provide the seminar and talk with some of our faculty here. Noah, any last words?
No, but thank you so much for joining us and speaking to us. It was a really wonderful talk.
Great. Thanks. Thanks again for the invitation. Take care, folks.
Thank you.
Oh, yes, and go to you.